Okay, great. So I am thrilled to be hosting Duygu Ucar today. And in reading through her CV, I realized that we've had kind of parallel trajectories a bit here. So I mean, a little bit, right? So she did her bachelor's in computer science. I did mine in math, and then she was an NSF Computing Innovation Fellow and a postdoc from 2010 to 2013 at Stanford. I did my postdoc at Berkeley. I had an NSF, right? I was like, wait a minute, whose CV am I looking at here? And then she was in Denmark in 2008. And believe you me, I was in Denmark in 2008.
No
What?
way.
No way. How did we never meet?
Oh,
And
we haven't met in person. We have to fix that.
we have just been crisscrossing the literal globe. And it's just wild. And so in 2013, she started as an assistant professor at the Jackson Labs. And I believe we met in 2020.
Oh, yeah,
Um,
you were visiting, yeah.
yeah, and then she was promoted to associate professor there at Jackson Labs in 2019, and just does some really cool research. In our lab, we read some papers in preparation for this, and I just want to highlight as someone who's interested in sex differences. So we don't think about aging as much. I know that a lot of Dr. Uchar's research is in aging. But we find that people often don't design experiments to consider sex as a biological variable really well. In this paper, I was just like, look at these sample sizes. Look at all of that. Look at that design. This is so great. So you are in for just like a wildly exciting and just scientifically stable and incredible presentation today. So with that, I will let you go.
Okay, Melissa, thank you for the kind introduction and kind invitation. I am truly excited about this too. So I think this can bring hopefully a lot of joint projects and cool collaborations together. So that would be wonderful. So feel free to stop me anytime. I don't see most of you. But if you have questions, just raise your voice and we will stop, okay? So I just wanted to give you a summary of some of the research going on in my lab, especially related to sex differences in immune aging and vaccine responses, which is an area that we are particularly excited about. So the approach that my lab is taking, as Melissa nicely summarized, my background is more computational and genomics. So we are taking a more systems level approach by recruiting donors. And most of our studies are using blood samples. So it is an easy to assess tissue type or sample type. So we recruit donors in collaboration with our collaborators. We work closely with the Aging Center or with clinicians at different institutes. So we use the next generation of single cell nowadays assays to profile these donors and combine it with a lot of omics, computational genomics analysis. So in this area, some topics that we are particularly focusing on is uncovering how transcriptional regulatory programs of immune cells change with age, and what's the role of biological sex and sex hormones in immune system aging and immune responses. And finally, how does both aging and biological sex affect vaccine responses? I'll give you an example of this towards the end of my talk. So just in case, and I know there are undergrads in the room, so some basics of the immune system. So immune system has two big arms. So the first defense whenever your body is encountering a pathogen is the innate immunity. So this could be a virus, bacteria, parasite. So the first response comes from these cells like macrophages, dendritic cells, neutrophils, which are fast in responding, but they are non-specific. So they mount similar responses to all of the different pathogens. So when these cells are activated, a subset of them, antigen-presenting cells, particularly dendritic cells, can move to the lymph nodes and can carry on themselves as tiny portions of the antigens antigenic peptides. And these peptides are presented to the adaptive cells, the T cells particularly, which then activates the adaptive arm of the immunity, which is developing more slowly compared to the innate immunity, but it is a more specific immune response. So it's a specific response for, let's say, influenza virus versus SARS-CoV-2 virus. And what is more distinct between the two arms of the immunity is the adaptive immune cells develop a memory. So you have a long-lasting memory of these encounters. So with this very, very simplistic introduction to immune system, let me also further summarize what happens to the immune system with aging. So both innate and adaptive arms of the immune system goes through changes with aging. So in the innate immunity, we see increases in the monocytes and dendritic cells. So these are the cells that also give rise to inflammatory responses. And there is a systemic upregulation of the inflammatory responses. Some people call this inflammaging because it's age-related increase in inflammation. And there are some molecules, excess production of inflammatory cytokines, IL-6, although we don't really detect it very well in transcriptomic data, has been shown to increase systematically with aging. On the other end, we see a chronic inactivation of adaptive immune cells. So this starts with loss of naive T cells. So naive T cells are made in thymus. Thymus shrinks with age, particularly because of that. There is a loss of naive T cells. Naive T cells are the T cells that can recognize novel antigens. They have not seen any pathogen yet. They are not trained. That's why they are naive, right? And in addition to this decline, we also see an accumulation of dysfunctional cells, which have impaired responses and diminishes your immune system's ability to fight any pathogenic molecules. In addition, we also have a contraction in T and B cell repertoire diversity. This is the number of molecules your body has encountered and responded. It's kind of your immune memory, right? So your immune memory declines also with age. So together, these changes, both in the innate as well as the adaptive immunity, significantly reduces our ability to fight against diseases, starting with infectious diseases, but many other, especially age-associated diseases, have an immune component. More than that, we also see a decrease in responses to vaccination and therapy. So some therapies or vaccines that are very effective in younger adults stop working for older adults. And together that really shorten the health span and lifespan of donors. So one of the first studies we did in this area, and we asked the question about the genomic biomarkers of human immune system aging. And I'll first tell you about this study. So for these studies, aging studies, we have been always collaborating with the UConn Aging Center, led by George Kushel. He's the director of the Aging Center, and he sees older adults regularly. He's a geriatrician. So with George's team at the Aging Center, we recruited 77 healthy subjects, young and old. And these donors donated blood for our study. And this study was led by Eladio when he was still in my lab and with our close collaborator, Jacques Banchero. So from these blood samples, we typically generate flow cytometry data to see, because in your blood, we first isolate peripheral blood mononuclear cells, which includes both innate and adaptive cells. We also generate flow cytometry data to see cell compositional changes because there are significant changes that happen with age. In addition, we complement this with genomic assays. So both epigenomics, typically we use ATAC-seq for that, and RNA-seq for transcriptomic changes. And we do a lot of computational analysis to be able to integrate and properly analyze these data. So this is our, in a way, pipeline to get these data and generate and analyze these data. And we work closely with George's team to screen these donors, especially important for older adults. because we do not want donors who have any confounding diseases like autoimmune diseases or who are going through active infections or active immune-related treatments to be part of these studies, which will be a confounding factor, right? So this is our pipeline to be able to build these cohorts and analyze them. So in this first study where we studied data from 77 donors, one genomic region and genomic signature drove our attention. And this is around the IL-7 receptor gene, IL-7R. And initially, these studies, we have been using PBMC bulk RNA-seq. And this we discovered from the PBMCs, which again includes all different types of immune cells from your blood, B cells, T cells, monocytes, DCs, etc. So what we have observed, and this was a computational finding, although IL-7R is a very important molecule for immunology, we didn't really have this expectation from the beginning. By generating RNA-seq and ATAC-seq data from young and old donors and comparing them, we observed that this region is going through very significant changes with age. So I'm here showing you chromatin accessibility in young and old, as well as gene expression between young and old. So we observed that both there is a chromatin closing in the vicinity of this gene, some promoter regions and enhancer regions, accompanied by the decline in the gene expression levels. So IL-7R is an important molecule. It's a receptor expressed on the surface of T cells, both CD4 and CD8 T cells. And if you zoom into the IL-7 signaling pathway, this receptor, when activated by the IL-7 cytokine, activates the JAKs and STAT signaling pathway. And this is an important signaling pathway for many immune responses, including the interferon responses for antiviral responses. So what we have observed is when we looked at the whole signaling pathway and when we looked at what other molecules are affected by age-related epigenetic changes in this pathway, we noticed that not only the receptor that is expressed on the surface, a lot of the downstream molecules, JAKs, STATs, are also epigenetically modified with age, and their chromatin is closing with age, suggesting that the functionality of the signaling pathway is impaired. To further test this, we got blood samples from healthy young and healthy old donors, N equals 4 in this experiment. We sorted out their CD4 and CD8 T cells. These are distinct T cell subsets with distinct functionalities. Both of them are expressing IL-7R equally on their surface. And we stimulated both CD4s and CD8s with the IL-7 cytokine, which should trigger this pathway. And then we looked at the phosphorylation of STAT5, which is the downstream in this pathway. And this analysis, this functional analysis, showed us that it's not the CD4 T cells that is stopping to respond to this IL-7 cytokine treatment. It is really the CD8 T cells. So this decline that we see at the PBMC level from our genomic assays, we eventually managed to link it to CD8 T cells and to this signaling pathway defects in responses of CD8 T cells. So that was our first study, and we learned quite a bit in that study by just studying the blood samples from young and old donors. Then I got really intrigued by the question of sex differences, which is, to my opinion, very understudied, and we can definitely do a better job here. So to be able to properly study this, you have to build a good cohort, and you really need to make sure that you are recruiting the same number of men and women, but they also need to be clinically comparable. So it's not an easy business. And probably for this reason, many studies either report no differences or don't even care. But we did care. And what we then did, we went back to our collaborator, George Kushel, and asked his team to complement our cohort and make sure that we have enough number of men and women for meaningful comparative studies. Our initial cohort was mostly women. not by design. Usually women, at least in our area, volunteer more for these studies. So we really work closely with the aging center and try to recruit men who are matching our existing women cohort, both in terms of distribution of age, but in terms of like frailty score, which is a clinical score we use for their overall health, any confounding diseases, BMI, etc. So of course it's a human study, so it's not perfect, but we try to match these cohorts as good as possible with our collaborators. So at the end of the day, we had 81 men and 91 women who enrolled in this study. And here we also managed to include middle-aged donors from 40 to 65, in addition to young and older donors that we studied in the first study. So again, we apply the same pipeline. We collect blood from these donors, isolate PBMCs, do genomic profiling in addition to flow cytometry to carefully study cell compositional changes and the computational analysis. So what was very striking from the beginning from this comparative analysis was, of course, we looked at our favorite molecule now, IL-7R, but we observe both in gene expression as well as chromatin accessibility level. Although both females and males are losing this molecule with age, the loss is more significant in men in terms of magnitude, which we observe both in changes in chromatin accessibility, which you can appreciate in this figure, as well as the expression levels were declining much faster in men. And IL-7R was not the only molecule. So if we just looked at when you compare old and young donors, in the ataxic space, we identify differential peaks. It's like differentially expressed genes, but more chromatin accessibility changes. So when we did this analysis in females comparing young and old and males comparing young and old, we were surprised by the fact that although the cohort was comparable, we had twice as many peaks, 10,000 peaks associated to aging in men compared to the women. So this was all done in bulk. Now we are repeating this in single cells, and I'll show you some slides from that. But to be able to deconvolute this bulk data, Eladio established a nice pipeline. Some of you might be familiar. There is a big human epigenomics consortia projects called Roadmap Epigenomics, where they profiled many human cells and tissues using different types of epigenetic profiles. And Eladio got all the immune-related signatures from this and compared to our opening and closing ataxic peaks with age, which gave us this really nice summary. So what we are looking at here is both in men and women, there is an activation, orange is opening with age. There is an activation of innate cells, which is in alignment with what we know from the literature. With age, we know there's an activation of innate cells, as I told you at the beginning. And there is an overall inactivation of the adaptive cell-related epigenetic regions. What was more surprising to us, the changes, the difference, sex differences in the magnitudes of changes. Although both, for example, if we just focus on CD14 monocytes, although both men and women experience this chromatin activation with age in their PBMCs, if you look at the number of peaks we detect in men versus women, this was a significant difference between men and women. So there was even more activation in men compared to the women in these CD14 monocytes related regions. So we then looked at the literature. There are some studies, especially this one is from Europe 500 Functional Genomics Project, where they recruited and studied more donors. So we found this cohort and their data where they looked at particularly pro-inflammatory cytokines in the plasma serum of these donors using ELISA. So what we then did is took older adults from this separate cohort from Europe, especially focusing on older than 65, that is the definition of the age cutoff we use for our studies. And then we compare in their plasma the level of these cytokines or the receptor antagonists for these cytokines. And this includes IL-6, which is frequently mentioned in the context of inflammaging. What we have observed from these cytokines and this analysis is the fact that as our genomic data has predicted, there is more pro-inflammatory signatures in the blood of older men compared to older women from this separate cohort. Nowadays, what we are doing, this is not published yet, but it's a work in progress. Nowadays, we are repeating these analysis and observations using single cell assay. So we have a beautiful cohort. We have blood samples from 96 donors that spans the whole lifespan of humans' life. So we have infants starting with a few-month-old infants, children, young adults, and older adults, including 90+ years old donors. So what we are doing now in this cohort of almost 100 people, we have already collected PBMCs. We are generating single-cell RNA-seq. We have generated single-cell RNA-seq, and we are studying, we also generated single nucleus attack-seq. We are looking at single-cell resolution, the effect of age, and also the sex differences in each of these age groups. I'm going to admit someone. Give me a second. Okay, done. All right. So just to give you an idea about the magnitude of this. So this is from, again, 95 donors. We have more than 600K cells from these donors. And you can see now, initially what we were lumping together as PBMCs have so many different cell types in it. We have B cell subsets, we have monocyte subsets, DC subsets, even a tiny population of stem cells, T cells, B cells, NK cells, you name it. So we are now looking at it in great resolution, which teaches us even more. So one thing that I told you at the beginning of the talk is this increase in innate immunity with age, right? So we can now see very clearly in these cohorts how this looks. So we are just looking at CD14 monocytes. This is the percent of CD14 monocytes within the PBMCs in different age groups. And you can see how the textbook description matches the data. You have very little of these cells when you are an infant. It gets more as you get older and older, and older adults have definitely the most number and most percentages of these cells in their blood. So what we then did to understand whether these accelerated innate immunity aging phenotype holds true in the single cell data, we zoomed in and compared different subsets of myeloid cells and the percentages in old females and old males from this cohort of 95 donors. And what we have observed, just like in our bulk data suggested, what we have observed is that there are more myeloid cells in old males compared to old females. So this was very obvious in CD14 monocytes as well as conventional dendritic cells. We also zoomed in and looked at some of the pro-inflammatory molecules, the expression level of some of the pro-inflammatory molecules in these cells. Here I'm showing you the heat map from CD14 monocytes. You can appreciate the fact that with aging, there is an increased expression of these molecules. That is obvious. But this is much more significant in male samples with respect to female samples. So what we have observed from the box study that there is probably an accelerated inflammaging in men holds true with increased resolution from our single cell study to which we are preparing for submission. What also Eladio observed from his study, when we compared, I told you the aging patterns kind of diverge with time between men and women, which was surprising to us. To further understand this, we compared ATAC-seq and RNA-seq data from older men and older women. And we observed that older men have more innate bias, as our single cell data also suggested, whereas older women have more adaptive bias, which might also explain some of the frequency of autoimmune diseases in women compared to men. So we did further analysis to associate these two pathways, including IL-7 signaling as well as inflammatory pathways. And at the end of the day, we observed that although the trends of aging are similar, the magnitude is really distinct between men and women. And this slide is, again, oversimplifying a complex story, but summarizes what we are seeing here. So what we have observed is that in the case of innate immune aging and inflammation, yes, both sexes experience an activation of this, but the magnitude of activation is much stronger in men. And adaptive cells, yes, especially T cells, both sexes experience this, but this is much more significant in men compared to the women. I didn't have time to talk about it, but since we have the Middle Ages, we also did some analysis, computational analysis to establish when these genomic changes start in men and women. And that also gave us some interesting differences. And we call these breakpoints where genomic changes really accelerate. So the breakpoint in men were happening 5 years earlier than in women. And that's striking because that's roughly the lifespan difference we expect in United States between men and women. So another interesting thing what we observed from this data was the fact that male and female blood immune cells diverge after 65. Older women had more active adaptive immunity. Older men had more active innate immunity. This is critical because if you go to the doctor, sometimes, not always, sometimes they take your age into consideration when they give you a vaccine, for example, but sex is almost always ignored, right? I think we need to change that. And our data suggests a good reasoning, scientific reasoning for why this is critical to change. And the big picture, of course, and I'll try to focus on this in the second half of my talk, is the implications for health and lifespan. So very briefly, we have many reasons to further understand sex differences in health span and lifespan, starting with life expectancy, because in the US, this difference, as I told you, is 5 years. But if you look at across the globe, in every country, to my knowledge, women live longer and significantly longer than men. So there is definitely a lifespan advantage of women over men. But it doesn't stop there. So there are also differences, sex differences in immune diseases. Some diseases, especially infectious diseases, COVID-19 was a good example, are more severe in men. More men lost their life, unfortunately, to COVID-19 compared to women. But autoimmune diseases are more highly observed in women, especially lupus. Lupus is a good example. 90% of lupus patients are women. What is even less known is sex differences in immune responses. And this is an area that my lab has started to work on. And I will show you some of our findings from a recent study in the rest of my presentation. So in this study, we were really interested in understanding pneumococcal disease. This is bacterial infections. There are more than 90 strains of bacteria that can give rise to these infections. And typically, they are more severe than viral infections and viral pneumonia. Some of these statistics are before COVID, but 500,000 people are hospitalized and thousands of people die from bacterial pneumonia every year in the US. And most of these are older adults, as you can imagine. So there are vaccines available, especially after 65 years old. These are recommended, but the efficacy of the vaccines also decline with age, just like any other vaccines. And in the older adult population, this ranges from 50 to 70% based on the vaccine type. So there are two, at least when we started this study, which was before COVID, there were two major types of bacterial pneumonia vaccines available for older adults. And these are all including bacterial serotypes. One is called pneumovax. This is the Merck vaccine. The basic structure of this is basically containing the bacterial serotypes for 23 different strains. That's why the name is PPSV23. And this is mainly these bacterial serotypes are binding to the B-cell receptors and activating the B-cell receptors. So this is mainly inducing a B-cell response. The other alternative is a conjugated vaccine. It's a conjugated vaccine because the bacterial serotypes, in this case, you have 13 serotypes. Bacterial serotypes are conjugated to a protein called CRM197, which can activate the T cells in addition to the B cell activation via the bacterial serotypes. And also another difference, this has an adjuvant. Adjuvants are ingredients that are included into the vaccines to increase the immune responses. And we are seeing more and more adjuvanted vaccines, including for flu these days. Anyway, so we had these two different types of vaccines, conjugated, unconjugated. But what we didn't know is side by side, how do the immune cells of older adults respond to these two types of vaccines? And more importantly, who will benefit more from which vaccine, and can we learn this by doing genomic assays on this cohort? And this is also termed as precision vaccinology, just like precision medicine. Instead of randomly giving people whatever vaccine is available, we try to understand who responds better to each vaccine and then tailor their treatments and vaccination strategies based on that. All right, so I already introduced the vaccines a little bit. So this T-cell independent, it is called T-cell independent from my studies. It's not completely true, but bear with me. So this is more triggering the B cells because these polysaccharides from the bacteria is binding to the B cell receptors and activating the B cells. On the other hand, we have this alternative, which includes the CRM protein, where it is directly activating the T cells in addition to the B cells activation through the polysaccharides. So current CDC, this keeps changing. I cannot keep track of CDC recommendations for this vaccine, honestly. But at least the last time I checked, it was because there are newer versions of this conjugated vaccine that includes more serotypes. The latest recommendation is to give these. I think if you get PCV20, you don't get pneumovax a year later, but if you get PCV15, you still get the pneumovax 1 year later. But this keeps changing very frequently, so don't quote me on this one, okay? So what we have done in this study, again, working with George and Jacques, we recruited older adults, older than 60 years old for this study, who are naive to this vaccine. They never got this vaccine. These are not yearly vaccines. You don't get them every year. This is usually, depends on the vaccine type, but it's usually every 10 years or every 5 years. So what we did is recruited this cohort, 39 donors, who are naive to the vaccine. They never got the vaccine, and to their knowledge, they never got the disease. So this would be their first immunization for this bacterial serotypes, which is critical because it helps us to understand who are really the true responders and non-responders to vaccination. Anyway, half of our cohort got the Prevnar vaccine conjugated, the other half got the pneumovax vaccine. And again, because this is important to us, we try to recruit the same number of men and women to each arm of the study and try to balance them both in terms of BMI, age distribution, and other clinical measures important to us. So in these vaccine studies, we have a longitudinal design for a reason, because every time point have a meaning for their immune responses. Baseline before vaccination is important because we would like to understand, is there a baseline immune signature from this cohort that can teach us anything about their future responses. So baseline is really critical. And also we look at their baseline antibody titer levels and day 28 antibody titer levels to see who is responding, who's developing these antibodies and who is not. Day 1 is critical because that's when you see the innate responses. I told you innate immune cells are activated first. So day 1 is the peak of the innate immune responses. And day 7 to 10 is critical for adaptive responses. In this study, for logistical reasons, we had to do day 10, but honestly, it was a little bit late. So then we usually have a later time point to see if the immune cells are going back to the baseline and if there is any memory cells associated to the responses, etc. So the quantification of this vaccine is a little different than flu vaccines. For this, we work with Munnam at University of Alabama. He's a world-known expert in this area. So for this, we use opsonophagocytosis assays, which are more functional than ELISA that we use for flu. It's basically looking at putting the phagocytes, which are cells that can eat bacteria, just putting the serum from donors, and the phagocytes can eat the bacteria faster if it is covered with antibodies. So you just look at how much killing is happening before and after vaccination by using the serum antibodies. And if the killing has increased, then you know that person has a lot more antibodies, right? So that's the basic idea behind it. And Munnam did all of these for us. So what we have done is, sorry, I need to see. We cumulatively looked at, so because one vaccine has 13 serotype in it, the other one is 23, 13 of them are shared. So we quantify the responses of these 13 shared serotypes. And these are the most important ones, and that's why they are used in PCV13 vaccine, because they are the ones that circulate the most in the community. So when we compare, so these are showing antibody cumulative from these 13 serotypes, antibody levels pre and post. If you have a good vaccine, you want to see an increase. And for both vaccines, we see a significant increase pre and post. So donors are responding to these vaccines. That's great. There is a slightly better response to Prevnar, the conjugated versus Pneumovax, but they are both inducing strong responses. What was more striking to us was the individual level heterogeneity. Although as a cohort, they are both responding, you can see here. So at the donor level, some are somewhat responding, some are really well responding, and there is all this heterogeneity, which is interesting to study. And to be able to study that, we derive some computational measures to rank the donors in terms of their responsiveness. So I'm going to walk you through these analysis. Here we are looking at the 13 serotypes that are quantified for vaccine responsiveness, right? And every row is a donor and the sex and age of the donor, etc. So these dots are showing the fold change from pre and post vaccination. So if these dots are bigger, it means for that serotype particularly, there is a strong response. So ideally, you want to respond to every serotype. But if you look at the donor level, there is a lot of heterogeneity. There are some donors who respond to most of the serotypes. There are some donors who respond to only a few. So we came up with some ways to combine these data and rank the donors. Higher rank means your responses are really strong. Lower rank means your responses are weaker. And then we did compare top responders, the donors who give really good response, and the bottom responders, the ones who did not respond as much. And of course, one interesting thing is age. Although everybody is 60 and older, there is still a 20-year window here. My collaborator, George Kushal, told me that 60-year-old is considered the pediatric arm of the geriatric unit, which is really funny, but I wanted to share it with you. Anyway, so what we observe, especially for Prevnar, that induces T-cell responses more directly, age was a statistically significant association. So if you are in your 60s, you will benefit more from this vaccine if you are in your 80s. But for the other vaccine that is B-cell related, we did see a trend, but it was not statistically significant, which is interesting because in our other studies too, we see that B-cells age much more differently than T-cells, and T-cells go through much more significant changes than B-cells with aging. That could be why one vaccine is doing a better job in older adults than the other one. Anyway, what was even more surprising to us was the sex differences. When we compared the responses to the T-dependent vaccine PCV13, we observed that women mount stronger responses compared to men, which was not the case for the other vaccines. So this is, again, very vaccine specific. That's why it's important to build these cohorts and study the sex differences vaccine by vaccine. Then we start looking into baseline predictors of vaccine responsiveness. The idea here is we already know who are responders and good responders and not good responders, right? And we have collected blood before vaccination from these donors, and we profile their immune cells extensively prior to vaccination using flow cytometry, RNA-seq, any other assays you can think of, we do this. So I'll show you some data from the bulk RNA-seq pre-vaccination and how informative this could be. So this is Satya's project. She built a pipeline using the WGCNA algorithm, where we look at the bulk PBMC RNA-seq data from donors. And then using WGCNA, she uncovered 32 modules, gene sets that are co-expressed. And she correlated these expression of these gene sets to rank responsiveness to Prevnar and responsiveness to Pneumovax. And we uncovered two distinct gene sets, one negatively associated with Prevnar responses, and another one, a distinct one, negatively associated with the other vaccines responses. The Prevnar responses, when we zoomed into this gene set, we were very intrigued because this gene set included really important molecules for cytotoxic immune cells. So this is your natural killer cells as well as CD8 T cell subsets. So in short, we call this the cytox module because it includes a lot of molecules related to cytotoxic cells as well as the cytotoxicity function, including NCAM1, granulosin, perforin, etc. And if you look at the expression of these genes prior to vaccination and their responsiveness to vaccination, you can see how beautifully this can predict if a donor is going to benefit from this vaccine or not. So here I'm showing you the top 3 genes from the cytox module. So NCAM1, also known as CD56, is very critical for NK cells. And if you just look at the expression of this molecule in this cohort, you can pretty much guess if this person is going to respond to this vaccine or not, this Prevnar vaccine or not. And what was very striking was this was vaccine specific, which is important because if you have high expression of NCAM1, you are not going to benefit from Prevnar, but you might still benefit from that other vaccine. So that's why it's beautiful that this is really vaccine specific. And then to understand where this gene expression signature is stemming from, we take the baseline PBMCs from top responders and bottom non-responders and generated single-cell RNA-seq data, which is here shown on the right. And we first looked at where the cytox gene expression is highly expressed. As we expected, this is coming from NK cells and some of the CD8 subsets. And then we zoomed into the NK cells. In the blood, you capture mature NK cells, CD16 positive, and immature NK cells. We noticed that they have different functions. We'll come to that, but what we observe is that these differences between responders and non-responders are really coming from the CD16 positive NK cells. So non-responders had more of these cells compared to the responders. And that was one of the single cell signatures that we observed. This CD16 positive NK cells, they are more cytotoxic in general, and they have the antibody dependent cytotoxicity function. So because they express this molecule CD16 on their surface, they can talk to the B cells and interact with antibodies and induce killing in the target cells covered with antibodies via this function. Normally, these cells are good for immune responses, but what our data suggested is that maybe it's not always the case. And then what we did is we zoomed into these CD16 NK cells from responders and non-responders, and we compared their gene expression profiles from the single cell data. And we observed that non-responders have higher expression of some critical molecules. They look more cytotoxic, they look more activated, they have more interferon gamma expression. So together, these data suggested that non-responders, not only they have more of these cells, but their cells are more cytotoxic and more activated compared to the responders. And I told you this vaccine, there were sex differences and women respond better. This is that figure here to summarize that. And one reason, potential reason why this is happening is because men and women have different levels of cytox activity. So here I'm showing you from our cohort NCAM1 perforin granulosin expression. And all of these molecules, this is bulk transcriptome, are more highly expressed in men. And similarly, if you look at the CD16 NK subsets that we learned from single-cell RNA-seq analysis, men have more of these, which is detrimental to vaccine responses compared to women. So we don't know why men have more cytox activity, but that is the reason why they do not respond as well to this vaccine. So the other type of analysis we did to learn the baseline predictors is generate flow cytometry data before vaccination and compare the percentages of different cell types to vaccine responsiveness. So we looked at different subsets of dendritic cells. There was no association with vaccine responsiveness. Different subsets of B cells, there was no association, significant association with vaccine responsiveness, and then CD4 subsets. So here we observe that Th1 and Th17 cells are important for Prevnar responses. And having more Th1 and having less Th17, which are pro-inflammatory, are good for vaccine responses. So if you have more Th1s, which do a lot of killing, and if you have less Th17, which are more pro-inflammatory, response-inducing, you will do better with Prevnar vaccine. And again, this was not the case for the other vaccine, which is good news because that can help donors who have this phenotype to benefit from the other vaccine. Which is striking, and we are building a study to mechanistically understand this with Silke Post, who's an NK expert, is this incredible association, correlation between this CD16 NK population and Th1 and Th17 cells. These are the three populations that predicts vaccine responsiveness for Prevnar, right? And if you look at the donor level percentages of these cells, they are highly correlated, suggesting that they are not necessarily independent from each other and that there might be a potential regulatory role for NK cells to affect Th1 and Th17 differentiation, but also their responses. This has been studied in mice. The first paper published in this area was in 2012, sorry. So this was a Nature paper where they have shown that, in fact, NK cells can modulate antiviral responses of T cells. This is not known in human, and we are collaborating with Silke Post to study this in human. Stay tuned on that. So to summarize this part, so by recruiting older adults, vaccinating them with 2 different types of bacterial pneumonia vaccines, we observe that the immune protection is in fact split. So there are some donors who have really low responses and some donors who have really high responses to each vaccine. And by comparing the good responders and the bad responders, we can learn a lot about what type of immune features affect responsiveness to each of these vaccines. And that's what we discovered in the study. What we observe is that the baseline predictors of Pneumovax and Prevnar are completely distinct from each other. I presented to you the Prevnar signature today. You can read about Pneumovax in our paper, which is published in January this year. So what we observe is that there is probably an NK and T cell association that diminishes responsiveness to this vaccine, and we can very well predict who is going to benefit from this vaccine. And interestingly, and maybe more interesting for this group, is there is a sex difference for this vaccine. Women benefit more from this vaccine because they don't have as much cytotoxic activity as men in this cohort. So that summarizes this new study. And why is this critical to uncover these baseline immune states or signatures? Because currently the way these vaccinations are prescribed is really just, there isn't much thought given into personalizing these suggestions, right? So sometimes age is considered, but not always. Sex is, to my knowledge, never considered other than HPV vaccines. So this is important for personalization because we can establish very simple tests to look at a few molecules or to look at a few immune cell types in your blood and very well predict who is going to respond to which vaccine. At least it was the case for this study. So that's why this can save time, this can save life, this can save money. So it's critical. But it's also important for us to understand what defect in your immune cells prevent you from taking advantage of these treatments. Because once we uncover this, we can think about ways to modify your baseline state. This could be with pharmacological interventions or lifestyle changes or maybe epigenetic modifications. But that gives us an opportunity to specifically target this pathway or this cell type, right? Or to adapt the vaccine. So if we know that one adjuvant or one conjugate protein is really affecting this type of immune cells, maybe we edit more or edit less or have alternatives to the existing vaccines to be able to rejuvenate immune responses of older adults. And this is a recent review we wrote on this topic to summarize what kind of baseline immune states have been associated to different vaccines so far, and why is this critical to study this. And we have an ongoing study. I don't have anything to show on this, but I also don't have time, but we have an ongoing study funded by NIH to do this for influenza vaccines. There are different types of it. We will apply the same pipeline here and try to uncover whether we can do similar personalization, precision vaccinology strategies for influenza vaccines. And I would like to take this opportunity to thank my lab. I have a really fun team. Very interdisciplinary, and as well as our collaborators. So we have wonderful collaborators, starting with George and Jacques, Adolfo and Moon has been helping us to quantify vaccine responses, and many others have been working with us and teaching us a lot of interesting biology. Thank you for your time. I think we have time for questions. And I'm happy to stay longer if you have more questions.
Yay. Thank you.
Thank you.
Yeah, maybe I think I saw Mason, you might have had a question. Do you want to kick us off?
Yeah, I was gonna ask if do you think we can expect some of your findings here to like extrapolate to mRNA-based vaccines and all that? Or do you think
Question. We have a small cohort, 33 donors, who got different types of COVID vaccines. So some of them got mRNA vaccines, both Moderna and Pfizer. Some of them got the J&J vaccine. So the responsiveness, this was quantified by Adolfo Garcia's lab. everybody, especially after the booster to mRNA vaccines, respond very strongly. So there isn't really that much difference, at least 1 month, because we usually do the responsiveness test 1 month. So it might be fading over time, but we don't have that data. So that's going to be a little tricky because mRNA vaccine responses are really strong. I think then we need to think a little bit differently than these vaccines, maybe not focus on the response in 1 month, but response in 6 months, etc., which we haven't done. But what we are doing then in that study is comparing different COVID vaccines, especially mRNA vaccines to J&J vaccine, to understand how immune responses differ between the different types of vaccines. And there are a lot of differences. Like in that study, we are still learning. And we are preparing a manuscript on that. Like NK cells are more activated with J&J vaccine, and some other cell types are more activated with the mRNA vaccine. So there are definitely differences. Responsiveness is a little more tricky for mRNA vaccines because initial responses are really strong for everyone, especially after the second dose.
Yeah, and I would assume there's probably not a sex difference in the uptake of the mRNA. It's probably about the same.
I don't know the literature, to be honest with you. Maybe
Oh, it's okay.
from others, but in our cohort, it's too small to even study that.
Questions in the room here? We got a question. Go for it, Baptiste. Yeah, it's that thing. It'll hear you.
Okay. Thank you very much for the talk. I was wondering, do you have any evidence that anti-inflammatory cytokines, IL-10 or anything, might also be increasing more in men, suggesting that there is just like a lot more of everything? And is this also linked to the responsiveness to the vaccines?
That's a good question. IL-10, we didn't specifically look for it. We are trying to establish a good assay to do plasma proteomics. We are working with a company called Genomics, but some of these cytokines aren't detected as robustly as we would like. IL-10 is expressed with an inflammatory response, but it's an anti-inflammatory molecule. I don't know. Maybe others have looked at it. So we did not add the plasma proteins to the pneumococcal vaccine study. I think there is a possibility that they could also be predictive, but we did not generate that data for that cohort. Plasma proteomics is a little tricky because if you look at Tony Viscaro's papers, he's from Stanford. Plasma is like a soup, right? Yes, immune cells from the blood contribute to that, but also you have a lot of tissues contributing to that soup. So we don't always see, this is to say that we don't always see a good association with plasma protein levels and the level of expression in blood-derived immune cells. But if I have to guess, I would guess that, yes, men would probably have more of these, but we did not test that.
I have a question. First of all, this is really phenomenal. It's great to see this cool work. And everyone in my lab who's studying aging has read in detail your 2020 paper, the Nature Communications. Actually,
that makes my day.
Yeah. And we used it to inform lots of stuff that they're doing. So it's really, really cool. I have a lot of questions. I'll just ask one of them though, which is a broader one about your like exclusion criteria and what that means for studying the aging response in a natural population, let's say, right? So you're excluding based on disease, current infection, meds, and frailty, right? Which seems like these are like abnormally healthy agers.
That's a good question, but we are not excluding for any disease. We are definitely not excluding for chronic aging diseases. We are excluding for autoimmune diseases because we know their immune responses are distinct. That's a very good question. So these donors are the major criteria we exclude for: do they have an immune disorder or are they on an immune therapy? we exclude for that. We don't exclude for aging diseases. There are some folks who have cancer in these cohorts. Healthy is kind of a misnomer for older adults because they have the aging diseases. But what we don't want is donors who have an active immune disorder or going through an active therapy, immune-related therapy that might affect their responses. And for the vaccine studies, our exclusion, in addition to these, our exclusion criteria is for them to not have another vaccine 2 weeks prior and 2 weeks after these vaccines, because your vaccine responses can be mixed together, right? So your responses to COVID vaccine and responses to influenza vaccine would mix together. So we don't want that. Other than that, we don't really exclude them, and they are community dwelling older adults. So they are really a good representation of healthy older adults who do not have significant immune disorders.
Thanks. I have like a page of questions here. I can take for an hour, but I'm not going to do that.
I think I
So
have.
no, she has a half an hour open after, and then there's some time later today. If you would like to, we can message them.
You send me a message. I can try.
Yeah.
That's good. And I'm happy to stay now. I have half an hour as necessary.
Or if you wanted to like come on, but I know I do want to give everyone a chance to like get on to their next thing. So it's 1 o'clock. So we should at a minimum formally close the session. And I'll stay on with you here for a minute at this link if that's all right.
Mm-hmm.
Well, thank you, everybody.
Thank you, everyone. This was a pleasure.